Welcome back, everyone. Hope you had a lovely break. It's only been about an hour and a half, so we'll save the workout, stand up, get the blood clots loose for maybe after lunch. Although you can do that during lunchtime. Shake it out, be ready to sit a bit. It's only another hour and 15 minutes of kind of material, if you will. So I think this is hopefully a useful bit of tutorial that I have to give you here, all in R. Again, for those of you who are planning to follow along, if you're having any technical difficulties, you can complain here in the chat. You can write in the Slack, write anywhere. Someone will help you with it. But just for the sake of kind of moving along, I'm just going to go through at a fairly even pace. I'm going to execute the code kind of along with you and try to explain it. Hopefully there are a couple of neat tidbits along the way that you can learn. And again, if you take one thing from it, if you take even a little bit of useful code or you learn a bit about how this works, then that's a mission accomplished. So I've got the screen shared here. I've made the font size 16. How's font size for this? Can everyone see fairly easily the code? I'm worried that I'm muted again. I'm not muted. I'm going to type in the chat. Akshay says it's good. Thanks, Akshay. Looks okay. Looks good. Everyone can see. Wonderful. Okay. So here's kind of a kind of stripped down, but essential and valid run at Similarity Network Fusion, with a couple of little bits built in. Binder link. Yes, we could get the binder link in there. Aaron, that would be very helpful. So we'll start off with the beginning. Also, this is R Markdown. We've done a lot in R Markdown. I'm not going to explain the whole format again. This doesn't look very pretty because it's kind of it's the code behind what generates a nicer knitted document. The HTML document is also available. So if you aren't following along with the code and running it yourself on Docker or Binder, then you could just look at the HTML document, which is also in the GitHub and has everything kind of laid out. You can see all of the words that are written here, and you can see all of the output from each one of these functions, and just kind of follow along that way. But it'll also be on screen here. So we're going to load a few packages here. To load the packages in our Markdown, again, in case you're just joining us for today, you can press the little it was a little play button here on the right side, and it turns to a red square when it's running. It runs all the code within this block. Typically in R, you can run it line by line. The nice thing about the Markdown documents is that you can cluster bits of code together. Don't worry about this little warning package associated. If you get this, it's really not a problem. I'm running this locally. You might not actually get this in the Docker. So the steps: updating the local repo from GitHub. So Aaron's kind of reminding you, if you're using Docker, pull the newest version of the Docker. So I don't know, they might have been updated with a few packages. I think these packages should still be there. At the very end, if we have time, I'm going to show kind of just a neat plotting device, like a network plotting tool in ggplot, so that you can look at, and it would work for variables too, but for visualizing clusters of people and visualizing networks, it's a nice kind of bit of code that you can even think about adopting into your own workflow. It helps visualize these types of analyses. There's lots of ways of doing it other ways of doing it in R, and it's quite popular ways of doing it in Python. This is the one that works best, I find. So the local, you update your local repo from the GitHub of the summer school. So this still is day 5. Most of this is going to be kind of repeated from last year, where I did a similar SNF tutorial, and with the addition of this kind of network plot at the end. So, spoiler alert. So the first step in your analysis, the premise we're basically starting with here is a data set that consists of, like a clinical data set that consists of lots of different data that we've captured on a number of people. And I'll show you what the data are in a second. It's a clinical sample of people. And just like we've been hearing, these examples of integrative research is very similar to that. We have genotype information for some of them. We have biological sex information. We have some EEG recordings for some of them. We have RNA sequencing from some of them. We'll say it's from their blood because we didn't take it from their brain, although here I have said prefrontal cortex. So perhaps this is based on a post-mortem data set. Actually, there are some data sets that are like this. The Common Mind Consortium data is one of them. So we may have post-mortem data from individuals as well as ante-mortem kind of recordings of EEG or epigenetic variation. So I'm going to read in now a data frame with this kind of block of code. And I'm going to call it pheno-long. And the reason why I'm going to do that is because it's a longitudinal data set. So there's a couple of points that I wanted to make with this workflow, which is that it's going to resemble a lot of what might happen when you get real data. The couple things that I'm going to deal with, one is formatting long and wide data formats and kind of how we think about long and wide data, and also missingness, having kind of missing observations more or less at random, and how we can cope with missing data. We can remove it, we can impute it. I'm actually going to run a version of imputation and tell you a little bit about it. But this block of code is going to do a couple of things. The first thing it's going to do is read in a data set, and it's a longitudinal data set. So I'm actually going to run it line by line, and because in RStudio you can see if I just read in this data set, you can see that there's 2,000 observations of 15 variables. And the first thing that's going to happen, and this will happen if you run this block of code, it will show us the head of both of these data frames. If we just look at the top of this data frame, head of phenotype long format, you'll notice that we have a column of ID, which is our subject ID, and this is what you get in epidemiological or medical data sets of humans. Each person usually has a unique identifier. But you'll notice that there's, subject 1 appears on 2 lines. And this is because this is a longitudinal data set. So this is what you may encounter as long format data. This is just one of the things that you should kind of get thinking about when you encounter study data. And here we have a variable called time. This could be month, this could be follow-up period, this could be whatever, but we have 0 and 1. So you have different states of some variables at different times. Other variables stay the same. For example, I have created 3 genotype of variables, which can only be one of 3 values, 1, 2, or 3. You can only have 3 genotypes at a given locus. A diagnosis that this person has. So all these people have a diagnosis of major depression, and that seems to be stable across time points. But I also have some variables here, imaging variables that have changed over time. So people were scanned, for example, at 2 different time points. You notice that their imaging changes. I just wanted to draw the distinction between long format and wide format data in this example. This is a reshape code, which I think Erin has updated. So this might look a bit different in the Docker because of one of the dependencies. There's lots of ways of doing this. I think she might have used tidyverse. But this data can be represented in a couple of different ways, and that's the point I wanted to make with this. After a reshape, where I say direction wide, I'm taking the exact same data frame. You remember that 2 IDs were on the same line here. I wonder if I run this, it'll show both. I think it does. Yeah, we can switch between them. Here was the original data: subject 1, subject 2, subject 3. Right, 2 imaging measurements for them. Now in wide format, you can see that Geno 1, Geno 2, Geno 3, they were never different. Your genotype doesn't change over time. And in fact, I've specified it as an ID variable, one that doesn't change over time. But you see now the other variables, like image 1, has now 2 columns. So we have like image 1, image 2, 0. We can show all of the remaining columns. Image 1 also has a second variable. The way that they've kind of lined them up is different. The point is we've split one column into 2 columns in order to capture time point 1 and time point 2. So that's straightforward. So now each ID only occurs on one line. Subject 1 is here. their first observation for imaging variable 1, right, at time point 0 is here, and in another column over here, at time point 1, not time point 0, the next time point, their value is 6. You see, it's on the same line, and it has to do with how we can analyze data. So it's something to think about. Let's read in our high-dimensional data for network fusion. These are simulated data sets, kind of based on real data sets, with the exception of the EEG data. Actually, I think that's pretty much entirely made up. And let's take a look at what they look like. So this read RDS function is the kind of go-to function for reading in a binary formatted R object. It's really handy for saving. If you're used to working with R data frames and objects and saving them as CSV files, like comma separated or tab separated or text files, and reading them in and out that way, you should get used to using RDS, the RDS file extension, because it really compresses these data sets into a binary format. It takes up less space on your drive, and also you can read in the R object exactly as it was in your session. So this is an R object I'm reading in. It's not a table that I then coerce into another format. It's the R object as I saved it as a binary object outside in my computer. Really nice. So 3 data sets, and let's look at them. Here we have a expression data set and the EEG data set and the methylation data set, and we can look at how they're actually arranged, and they kind of match up with the way that some of these omic data types are often arranged. They are already transposed from what we kind of just saw in a data set where often each row is a different subject. In this case, you can see, for example, in the expression data set, each row is a gene. And each column has a number associated with it, 3406, 3407, 3408, 3410, and so on. Each one of these columns is a person. It's one of our individuals. It's a transposed table. And each row is a gene. You often see the gene expression data is almost always organized in this way with subjects and columns. We can look at the EEG data, and what do we have? Well, it looks like the same kind of people. So the identifiers are the same, subject 3406, subject 3407, subject 3408. And the variables here are EEG peak frequencies for different bands. And they have some values here. We can ignore the credibility of what those values could take up for now. And it's just showing the top 5 rows. There are many more rows here. We'll look at the number of features in a second. And then methylation. And DNA methylation is kind of indexed by these CPG sites, or these CG nucleotide pairs, on which methyl groups are kind of added or removed by cellular machinery. And this is the kind of intensity of methylation observed for these individuals. So you see, we have the same people. All the columns are the same, 3406, 3407, 3408, but our features are different. So we have 3 different 2-dimensional matrices of different data types, and we want to understand, according to each data type, how similar are our individuals, then look across data types and see, are there patterns that are consistent in these 3 data sets, create the fused network, and then cluster that network. Can we come up with these molecular and electrophysiologically generated subgroups of people who might have major depression, they might have cyclothymia, this is a clinical sample, and we have all this phenotype data that we gathered on them, we can merge in with it and try to figure out what these biological subgroups actually look like. So the first thing is we're going to generate these patient similarity networks and fuse the networks, so you might remember. First, we should know if there's missingness in the data, and I'm going to talk about missingness with this example of introducing a bit of missingness, how you can look for missingness, and how we might fix it. So it's a little bit of an aside. We're going back to this pheno-wide data frame, you remember, from the very first. So this is kind of the data that had a couple imaging variables and some cognitive variables. These aren't our omic data types, right? This is data that's collected on some of the same subjects. You notice the IDs are different. We're going to map them together. But the data are different. We're not going to use this pheno-wide data for our clustering. We're not going to use it for our network fusion analysis. We're going to use it after to try to figure out what the biological networks mean. And so we're going to use the wide, as I said, the wide format data. We can go back to the front, where each person occupies one row, right? And this data set right now, it's actually a perfect data set. There's no missing information. You see everyone came, subject 1 attended, well, every subject attended their first and second imaging data set, appointments. They also had cognitive tests done at time point 0, 3 different cognitive tests at time point 0, and everyone came back for their second time point in cognitive tests and had a score for their cognitive performance, right? So it's a complete data set. You're never going to see this in real life. So let's create a copy of this data set and call it PW for phenome-wide miss. And we're gonna put some random missingness in it. We're going to simulate missingness. So I'm going to run this block of code. All that's happening in here is we are identifying the numbers of rows in this data set, which is identical to the phenome-wide data set. So it's the number of people. And we know from here, we can just look up the environment, there's 1,000 rows. It's 1,000 people. Notice how it's exactly half the length of the long format data set, right? Because long format is still 1,000 individual people, but they were measured at 2 time points. When we made it wide, we increased the number of columns and halved the number of rows. It's the same data, it's just kind of reshaped. And then what we're doing is this little bit here kind of looks a little bit messy. You can pull it apart if you want. It's really quite simple. We're just basically looking across all of the rows of our data except for the first 3, because I don't want to introduce missingness into the IDs. We always know what the subject ID is. And in our case, and as is the case in most samples, you have a good measurement of age. It's not a lot of missing data for that. Biological sex, these kind of fundamental things are usually accounted for. But people don't show up to appointments. Sometimes imaging data is of low quality and has to be removed. Cognitive tests data might be invalidated for one reason or another. There's lots of reasons why those variables might be missing across visits. So it's just going across the columns and introducing randomly 20 observations, or it's the number of columns divided by 20. So it's like a fifth of observations we're going to remove, 5%. So the number of columns divided by 20. We're going to introduce into each column, differently for each column, random NA observations for 5% of the data set. And that's what I've done. So now we have a data set, you'll see PW miss. It's exactly the same as phenome-wide, same number of variables, same number of observations, except that we now have 5% missingness randomly across our variables, which is not great. So how do we just gauge that? What if you just got a new data set and you want to figure out what your missingness looks like? Well, a couple ways to do it. Number one is for a large data set, there's no really good way. Although I perhaps should update this. A very talented summer student of mine, Roberta Doling-Borum, who is, I think, on this call or on the session, has done very well with a Python package to visualize missingness patterns in very large data sets. To kind of get a succinct statistical picture of numbers of your missingness across your variables, this is one way of doing it. It's basically just simply looking across your variables, identifying how many are of missing, and then looking at a table of this missingness. So if I just show you what this looks like, we'll run this block of text. We have a table here that tells us that there's actually 2 outputs here. So the NA out object has gone across our entire data set and just identified in every column how many are missing. And we have 950 missing observations and 21,050 non-missing observations. Again, 1,000 people, 22 variables, that's what you get. And remember, we didn't introduce any missingness into ID, into sex, into diagnosis. No missing data. But this is a way, if you have dozens or even a few hundred variables and you want some numbers on how many values are missing, you can kind of quickly get a picture of that with a summary method like this, which is, there's nothing fancy going on here. It's just kind of operationalizing the R code to get a picture. We can very clearly see that every variable now, and this is not by mistake, we engineered this into the missingness, every variable has 50 missing observations. Now, it's not the same 50 observations that are missing. It's not the same 50 people that are missing this data. That's been done randomly. But there's a total of 50 missing in each sample. There are a lot of nice visualizations that are built into some R packages for visualizing missing patterns, but they don't scale up well. So in this data set, you have like 1,000 observations and 22 variables. It's kind of hard to see patterns. But if I just take this data set with some missingness, and I'm just going to take the first 50 rows, so the first 50 people and all 22 variables, and just run this function called MD pattern, which is like a missing data pattern. You see it kind of ran through a function. It spits out a table. It's a kind of matrix of missing patterns of which variables, 1, are there and which ones are not there, 0. And it gives a nice representation of the patterns of missingness. And they have different summary numbers on each axis to kind of show you that pattern of missingness. So you can see that the left y-axis over here, these numbers 25, 5, 2, 1, 1, is the number of missing observations for each data type in each row. And the right y-axis is the total number of missing observations in each row kind of together. So the way to think about this is how many, so it's kind of odd to think about. There are, I don't actually know where the 25 comes from. Each one of these rows you can see on the right-hand side, if we start with the right axis, is 0, 1, 1, 1, 2, 1, 1, 2, 2, 2. This is a product of the randomness of what we kind of threw in. So there's 1,000 people and we're only making 5% of the observations missing at random. So in some cases, there will only be 1 missing observation. In some cases, maybe it doubled up and there were 2. So for example, here you had COG 2 at visit 1 and SES, socioeconomic status, at visit 0 were both missing here for this individual. And so there were 2 kind of missing data points for that person across 2 different data types. However, within each data type, so for COG2 and for SES, there were only 1 missing value was kind of found per person. I actually find it kind of complicated, if you ask me. I don't know if this is ideal. Last year it made more sense, but I think this year, it happened quite recently, and for future future tutorials, I think I'll have Roberta go about it. We don't want to mix in Python. I think there's a good way to kind of go about visualizing the missingness patterns. That said, this representation, although the makers of this package have done fairly well to come up with some kind of some measurements of missingness across the sample. One of the reasons, first of all, why it's very hard to kind of interpret this is because it's a bias interpretation. So we've only taken the first 50 kind of rows of data, but the missingness was introduced across a much larger group of people. So it's the pattern, just by chance here, you have some variables like SES1 and image 2.1 that got no missingness. So the pattern doesn't really reflect the missingness that we put into it. But even still, there are kind of valiant efforts to do something that's kind of impossible to do without looking at numbers, which is why I kind of start with this. You need to be able to visualize numbers of missingness. There's other ways to kind of creatively do it, but visualizations like this always kind of fall short in some way. Going by variable, and there are other ways of doing this. This isn't meant to be a tutorial on missing data. I just kind of wanted to show you a couple ways of going at it. And so hopefully we've done that and not injected too much additional confusion into things. Again, it's a nice resource to look at, and I would encourage you to, if you have data of your own and you are trying to assess missingness, to attempt this this method of visualizing and see if it can make sense to you and if it kind of helps your cause of identifying missingness patterns. Of course, once we've identified that there's missingness and we put missingness in the data in the first place, we know there is, we have some options as to what we can or can't do with the modeling procedure. So we can either remove people who have missing values, right? In theory, I mean, you can't really learn anything from a subject that you haven't measured a particular data point on, right? And that's kind of true, except for the fact that, well, I mean, it is true, because the best we can do is try to estimate or guess what that person's response may have been based on other data we did collect about them. And that requires other data that we've collected, and it becomes dependent on those other data, even though there's always randomness associated with that guess. The reason why we might not want to just remove people who are missing some data and just kind of stick with what we actually have, what new information we have, is really boils down to statistical power and having large numbers of subjects in your study. So you need people to actually make inference and to give any kind of confidence to estimates that you might get out of your model. So, every parameter that you estimate in a model is going to have some uncertainty associated with it, and one of the best ways, probably the best way to boost confidence in your parameter estimate is to just increase the number of subjects that you have. And another part of that is kind of conceptual. I mean, it's the difference between a case study and a huge population level estimate. And all of the themes that we've been touching on, personal medicine and individualized and personal medicine, precision medicine, and kind of group-based or just one-size-fits-all medicine. You need a lot of people to estimate true parameters. You also need a lot of people to understand that there is variability among people. So we need subjects. We need data, not a new story. Here's one method of imputing. This is implemented through the MICE package, which is this multiple imputation through chained equations. Not going to go into too much detail on the method. This is the default method for this package, which is called predictive mean matching. You can think of imputation as, again, I don't really have slides on this, it's better done visually, but you can think of the idea of imputation, of guessing, right? So we know that 5% of the observations for, let's say, a cognitive task are now missing in our data set. But that person is not missing data for other things that we've measured, right? Maybe they're missing their value for cognitive test 3, but we have their value for cognitive test 1, we have their value for cognitive test 2, we have an imaging measurement on them, we know their sex, we know their age. Imputation is really a modeling exercise, and a lot of imputation algorithms are based on, John pointed out, they're based on regression, they're based on linear regression. And indeed, so is predictive mean matching. There are limitations to regression. For example, in an unconstrained sense, linear regression has the potential for extrapolation, the potential for yielding predictions that exist outside of the observed domain of data. So if you have data types like categorical data, where you can only take on a certain number of values, if you have a very limited age, if you're estimating age, for example, and you run a predictive algorithm, it might predict someone's age being like 150, just because of the way that the algorithm is formed in uncertainty. But of course, we know it's not possible. There's no one in existence who's 150. So imputations need to be constrained, right? The way that we fill in gaps in the data need to have some limitations. We can't simply develop a multiple regression based on everything else we've measured on a subject and try to fill in the gaps. So the way that predictive mean matching gets around this is to develop multiple regression models in a multivariate space for all of the variables that you're trying to impute. So one variable at a time, try to impute this one column, develop a multiple regression of that column that develops you get a set of beta parameters, a set of beta coefficients. And there's a distribution of beta coefficients because you're trying to predict that variable as a set of other variables in the data set that could be informative. And the distribution of those beta coefficients is actually, it kind of exists on some probability distribution, right? Those beta coefficients exist in some, as part of a greater space that can be pulled from a kind of posterior probability distribution of beta coefficients. So you're not just taking those beta coefficients from linear regression and saying, okay, plug in, all of the values that aren't missing for this person to predict the thing that was missing. You're actually pulling fake beta coefficients, these beta star coefficients, from this potential distribution in multivariate space of beta coefficients. So there's some random shuffling going on there. You're pulling out the contributions of these predictive variables out of thin air, not out of thin air, they're informed by what you observed. And then there's another step, which is actually so you're coming up with a prediction of what that might be based on these sampled beta parameters. And then you're not just filling in the gap with a new value, you're identifying a number of people in your actual data set who look like that, who are close to that value. And then you're taking the actual value that one of those people has. There's another random selection step. You select a random person from the set of kind of closest matches or nearest neighbors, and you're picking what an actual value in your data set and filling it in the gap. So it's kind of like a selective bootstrapping, because you're actually looking at data you have and values that exist in your data set and filling it in, but you're doing it based on a combination of regression and kind of random matching of observations. And what that does is it keeps your model constrained. So if you had a binary variable that could only be one thing or the other, or a set of diagnoses, for example, your imputation won't produce some value that makes no sense, something that you have not observed, or some experimental condition that didn't exist. You wouldn't really impute an experimental condition, so it's a bad example, but the age example I'll stick with. If your age range in your study is age 20 to 40, your algorithm won't randomly impute someone who's 50 or 60 or 100. It's constrained to those. So that's what's happening here. The algorithm is going through, I'm going to run this block of code, it's going into the data set, it's intelligently identifying the rows, not the rows, the columns, the variables that were missing, or had at least one missing value, and then it's performing this predictive mean matching to fill in those gaps. And then I'm taking the complete observations, kind of complete version of the imputed data set, and sticking it into new data. Now, it is multiple imputation. What's happening is, because there's randomness at each one of these steps, you can see I've specified a seed, because it's actually a pseudo-random randomization. There is randomization happening. Multiple imputation basically performs many imputations of the same individuals, the same missing data, and there are some methods that can be piggybacked onto multiple imputations. So kind of you're taking your missing data set, creating like 5, 10 more complete data sets that are all a little different but don't have missing data, and can funnel those into your actual modeling steps. We're not doing that. In this case, I'm just taking one iteration, max it one. I'm taking kind of one guess at random space with the seed, so it should be the same for everybody, to get a complete data set of new data. Now, it did spit out an error which said number of logged events, missing observations remaining equals 50. And so we're going to be like, whoa, I thought we were imputing data. I'm going to jump through this because this is just a quirk of this imputation algorithm. The algorithm won't impute variables that are completely collinear with one other variable. So by collinear, I mean they're perfectly correlated. They're perfectly informative of the other. And we can look at what happened here. That's because age was measured at 2 time points, right? At time point 0 and 1. But age is fixed. Age is completely dependent on time point. And remember, it's a wide data set. So it turns out that age 0 and age 1 are identical, because the time difference between follow-up in this fictitious data set is the same for everybody. So in this fictitious data set, it was 6 months apart. So this means if you knew someone's age at the variable age 0 at the start of the study, you know exactly what their age is going to be at age 1. And just plotting it out, you can see they're obviously perfectly correlated. So the algorithm just said, hey, look, do your own work. We only have to impute one of these, and maybe not even that. So there's no point in running an imputation. It's just for preserving time. It's just for efficiency. That's all. We haven't lost any information. So that's just imputation. That's kind of fun. If it is fun. One way of doing it, one way of approaching things. So it was never meant to be an imputation workshop. It's something that I wanted to flag for you. There's 2 things. Thinking about longitudinal data, long format and wide format data frames. Thinking about them as no loss of data, stretching them, squishing them, having 2 subjects, or having one subject rather occupy multiple rows versus having one row, by definition, being belonging to one subject, and kind of spreading out your measurements and variables over columns. And keeping that in mind, and also just being aware of missingness and having one kind of way of solving it. Predictive mean matching is actually one of the more sophisticated and commonly used methods out there. There are other options. You can also impute by median, which is done. You can just take the median of values that aren't missing for a given variable or for a given column and just make that the missing value. At least then you know that you will always have a value that is within your constrained range of values. That doesn't always work in circumstances where you have categorical data or other kinds of skewed, very heavily skewed data sets. It's not informative. So predictive mean matching works for skewed distributions, works for categorical variables, works for numeric variables. 2 thumbs up. So we're going to jump into SNF, and the SNF workflow is fairly clean and fairly straightforward. We've got about 39 minutes left, so we don't have to rush. I'm curious to know. It's been difficult to engage, especially on day 8 in the home stretch, and with everything we've been through and all the Zoom. I'm curious to know if people are following along, or how people are feeling. So I'm going to do a little bit of interaction here. I'm going to do a poll. The poll is going to be, are you running code right now? And I think I've made it visible. Oh, there. Okay, I think it says visible hidden. Okay, great. I've made a poll. So let's see. There's 31 people who are live right now. You don't know this, but Crowdcast lets me secretly activate all of your cameras, so I can see you right now. I can see all of you. I've got like a big screen up, and I can tell if you're working. I can tell if you're paying attention. I'm kidding. Probably not good for legal purposes. No, there's no way I can't see you, but I can see you intellectually. So let's see, see the hands. We've got 8 people who are kind of responding to the poll. Some people there's more people than I thought, a little more than half right now, 11 people. 12 people are responding, 7 people are running code. And also you can answer this on its face. Are you running code right now? If you're just running code for something else, you can answer yes. You don't have to be paying attention. This is a general question. 14 people have responded. It's good. This is great. Wow, so about 50-50. That's really good. I'm gonna create another poll. This one is a little more self-serving. Well, yeah, it's important. I've added another poll. So you're already in the poll box. I mean, you can just click it. So, let's be honest. Again, remember, I can't see you. So was my little blabber of about 10 minutes on missingness helpful? I will not be offended if you say no or a tiny bit. There's a lot of options I haven't given. No, that's great. Thanks, everybody. Okay, I won't kill it for next year. Maybe I'll just make it a bit better. I'll invite Roberta up to do it for me. Okay, so a lot of you are there. I appreciate the activity. Thank you for the one person who said no. Thank you. 6.7% no. I'll just treat that as missing data. If I impute that no, maybe it lies in more yes, kind of mean matching. But no, it's important. This is good. Objective feedback is really important. Okay. Thanks for that. Good to know everyone's awake, paying attention. I'm gonna forge ahead. We're gonna do the SNF. We're gonna take a look at the similarity networks. And Shreyas, you're welcome, Dan. Why do I get the feeling that Shreyas was the one that said it wasn't helpful? I'm getting a feeling out there. But I appreciate it. It's great. Okay, so let's jump into Similarity Network Fusion. At very least, you'll get to tell your colleagues and peers and friends that you worked on something called Similarity Network Fusion. Anytime fusion is in the title, you kind of feel like you're in Back to the Future or something. Like cold fusion device for how they fly into the future. They kind of outfit that DeLorean with a cold fusion reactor. So fusion is just cool. Deceptively simple. The first thing that we have to do is, so you remember we've read in the data. So we can do, quick just a reminder, I'll just zoom back up here to when we read in our multimodal data, right? Subjects are in the columns and the data features are in the rows. And we had 3 different data types: EEG, gene expression, and CpG methylation, right? DNA sequence methylation. 3 different omic data types. Let's find out how similar people are to each other. Let's look for groups. Let's see what we have. 3 things that we need to do for this method. This method is surprisingly robust. So in any method, and any modeling exercise, there are going to, almost any modeling exercise, there's going to be some kind of decisions that you have to make before you run the model. We heard a little bit about this, we've heard a lot about this in different contexts over the last week. A particular, in what's relevant for my research is, choosing parameters for polygenic risk score calculation. How many SNPs do you include? How do you deal with linkage disequilibrium? These are decisions you make. We often refer to these as hyperparameters, parameters of a model that dictate how it builds itself or how it functions or how the algorithm runs, as opposed to parameters that you might be trying to estimate that relate to the data itself or the structure of the underlying data. So there are here some kind of algorithmic parameters that we have to lay out. The first one is K, which is number of neighbors. So the way that this algorithm works, one of the ways in which it determines the similarity between people and the way that it updates similarities across data types is by determining, you can think of it similarly to like predictive mean matching. In a lot of ways it bears resemblance to imputation, looking for similar observations and identifying nearest neighbors. So you can think of this as kind of, if you're looking for how similar one person is to another, how similar do they have to be actually be considered similar? It sounds like a bit of a philosophical question, and it kind of is. What ends up happening is in really high dimensional space, and what I mean is a very large number of variables, you can end up in a situation where, and here's a way to think about multidimensional space. In one variable, right, if you have one variable, it can be represented on a line, a number line, right? And a two-dimensional space, if you have two variables, you need like Cartesian coordinates, right? You have like a two-dimensional, you've done this with graphing calculators in high school. You have an X and a Y axis. You can plot things in this two-dimensional space. Three-dimensional space, also you can have these, these spatial coordinates of a three-dimensional plot, like a cube. Things can be represented in a cube. And then you have these, hypercubes, and then I don't even know. I mean, John could probably talk about this all day in terms of dimensionality and derivatives on derivatives and derivatives and everything else. Every time we represent data in a higher dimensional space, we are abstracting, and we kind of lose our sense of how close or far apart things are. Right? It's very easy to identify how similar two observations are on a line. In two-dimensional space, it's also fairly intuitive. In three-dimensional space, you can also think about that traversing a distance, between two objects. But when you start going in 4, 5, 6, 7, 10,000, 1 million dimensional space, it actually becomes very difficult to understand how similar things are to each other. And what happens is when you start using kind of traditional distance metrics like Euclidean distance, which is like this kind of closest straight line distance between 2 points, when you expand into a super high dimensional space, that distance metric actually loses its meaning. And in a similar way, when you're determining kind of the number of individuals to base similarity on, so how similar or different are you to a group of individuals, two things to consider is number one, computational complexity, and two, what value are you getting comparing one person to other people in this crazy high dimensional space, because they may be deceptively close to each other or extremely far away from each other, and that can bias the algorithms. I don't know if that's entirely clear, but the point is ideally we want to put some constraint on how far the algorithm is looking outside of one person's kind of values for a given high dimensional data type to identify its nearest neighbors and determine how similar or how different it is for that person across data types. The second one is a hyperparameter, which is fairly abstract. It has to do with the scaling of similarities also during fusion. And then T is the number of iterations, which is the number of times that the networks are fused. It is an iterative process. It basically takes one data type and updates it using information from the other data types. And then it goes again and updates another data type with the other two data types, one of which has been updated, and it continues to kind of cycle through in iterations. I mentioned a couple things. Number one, the iterations converge. So there is you're not running a risk of changing iterations. There's no kind of local minima or maxima in this problem. you're not going to run into problems of choosing a hyperparameter and ending up with too few iterations or too many iterations. They demonstrated in this paper extensively with very few iterations the algorithm will always converge with a large number of simulations that they did. Very robust. Also for hyperparameter and neighbor selection, they gave these ranges, 0.3 to 0.8, 10 to 30, based on their simulation studies of mixing up different omic data types and introducing all kinds of variability into the data. The algorithm is incredibly robust to these types of parameters, how variance is scaled inside of the similarity kind of equation that updates each network, and then also the number of neighbors that you choose. So, long-winded way of going about this, but I'm just saying these are the things, these are the decisions you'll have to make, and it turns out, of course, you can explore. You could run the algorithm making some adjustments to this within the ranges that they suggest, but in the extensive supplementary data on the paper that presents the method, you will see that they've done this for you. And they find extremely small differences in the way that the algorithm performs with changes to these hyperparameters. So I got to run this block of code so that I'm saving these values as k, alpha, and t, and these will go into the actual SNF function. Second step is we're going to normalize the input data. First, we must make sure that we match the subject names. This is something that's consistent, regardless of what you're doing. SNF, any type of data combining, you're running a mixed model, you're running any model at all, but you have multiple data frames. You want to make sure that they're organized the same way, merging them. In this case, we do feed the data frames in as kind of three separate objects in a list. And so we have to make sure that the order of subjects is the same in each of these data types. We don't want to scramble it because the algorithm will assume that the first column is the same subject in all three data types. So that's basically what's happening here. I'm just taking the column names from the expression data set and reordering the columns of the methylation and EEG to match and writing over those objects. So now I know that the expression data set, the methylation data set, and the EEG data set have the same order of columns for subjects. And then I'm creating a list object that just stores the expression, the EEG, and the methylation data together in this sense. And also there's a transposition happening. So I wanted to make this point, I guess, instead of doing it for us. As I mentioned, the output of a lot of these omic data pipelines and other pipelines as well, I'm not sure if it's the same for EEG, for some packages that are used if you're extracting features from EEG recordings, but quite often the features are in the rows and the subjects are in the columns. In this case, the algorithm wants it the other way around, and we use T to just transpose it. So we're transposing. Now the subjects are in rows and the variables are in columns, and we've created this list object that stores that. We apply a standard normalization to the data, and it's not always necessary. In this case, our variables, I think, are already pretty much in similar scales. But what can happen is when you're looking at similarity measurements between people, depending on what your metric of distance is, right? If you have some measurements that are in have a very high average or a very large amount of variability, and others that are very small in nature. So if you're combining features that, gene expression is actually a decent example of that. Some genes are extremely highly expressed and some are very lowly expressed. And depending on the distance metric you're using, very large values can bias similarity to one individual or another. A variable might have a greater importance than you want it to have. In our case, we want all the variables to have a similar contribution to the to the similarity of our individuals. We don't want to have the absolute expression of a gene to weigh itself in. We may actually want that to be the case, but we don't want that to be the case here. So I'm going to run this through and we'll take a look at the final kind of data configuration, what it looks like. So this is what it was. This is the original gene expression data. Remember, genes in rows and subjects in columns. And then here, doesn't look quite as pretty. It's just the transposed version of it. So now the genes are in the top. 1, 2, 3, 4, 5 genes. And there are the subject IDs, 3406, 3407, 30408. It's an R console output. I guess I could have used like a tidy table print to print it out. But it's just the transpose, so you can see exactly what's going into the algorithm. The work that's being done here is it's all pretty stock standard from the package. So you don't really have to do any work here. And actually, there's going to be a gotcha at the end of this. There's a bit of code that I didn't put in the final script, which is a funny kind of aside, but will help you in your work. So we don't actually even have to go through all of these steps. What's happening here is we're using a distance, a function that's built in, this distance function. I think dist2 is from the base package. Dist2 has some defaults in terms of the format that it spits out. And we are, another thing you'll notice is that in this data set, it's not actually extremely highly dimensional. For the sake of interpretability, I didn't make these, and also for speed of running, I didn't make the expression data set with 10,000 genes. I didn't make the EEG data set with, I don't know, I guess there's only so much you could really blow up. They're not individual leads. There's only 25 variables in the EEG data set, 150 genes in the gene expression data set, and 85 methylation points in the methylation data set. It just helps us with visualization after the fact. But the point is that a lot of these kind of determining distances and everything, like we don't have to worry too much about some of the caveats that I mentioned earlier in terms of defining distance matrices. And I believe this is just spitting out Euclidean distance, the kind of default behavior of this dist2 function. So we're looping through the data list. Remember, the data list is just a list object that has now our 3 aligned transposed data types. And generating these subject-wise adjacency matrices, which are just networks of subjects, again, within each data set. So we are applying these, getting a distance matrix, and then we're getting the affinity matrix. So you could think of them as kind of inverse. Sometimes they are inverse. In this case, there are some details of how this affinity matrix is calculated that are specific to SNF. But you can just think of it in some cases as kind of the inverse, almost like 1 minus distance in some cases, depending on your metric. So the end result, the thing that we actually feed finally into the Similarity Network Fusion, you remember for that slide that I showed at the end of the last session, we've got 3 equally dimensional data frames that represent the number of individuals that have this data type. So there are 106 people in this data set. I told you we've got 25 variables for EEG, 150 genes in gene expression, and 85 methylation points in our high-dimensional omic data types. But what we're feeding into the algorithm are these subject similarity matrices, these affinity matrices, that have the dimension of subjects by subjects. And at every point in this matrix, and I'll show you what this looks like in the next step when we generate the fused network, in each of these, you have kind of an equal assessment for each pair of people in your data set of how similar they are according to each data type. Now let's construct the fused network. This is the money. This is the meat of it. This is really what's happening. And the gotcha will be funny in the end when I show you. But anyway, let's run the Similarity Network Fusion. Let's run this iterative updating of expression matrix with EEG matrix with methylation matrix. How similar are these people according to individual data types? Let's figure out how similar they are if we look at all of the data types together. So it's run. It's done. It's fast. It's not quite as fast if you have, again, tens of thousands, hundreds of thousands of variables. The method is designed to handle that level of data. But this updating step is actually not the hardest part of the algorithm. Usually it's the, it's generating the affinity matrices, the distance matrix, and calculating the affinity matrix. It usually takes more time than the fusion. This is what the fusion network looks like. Looks pretty similar, doesn't it, to what we've been looking at. In fact, this network W is the fused network. It is identical in dimension to each of the individual data sets. It's also 106 by 106. And look at the rows and columns. Those are our subject IDs, right? There's 3406, 3407, 3408, 3410, 3411. They're the same people, okay? The way that this similarity is scaled means that a value of 0.5 is a identical observation. Obviously, subject 3406 is identical to observation 3406. They are one and the same. And then you can see the scale for the remaining similarities is all much smaller. 0.005, 0.01. Ooh, there's, look, this kind of higher than some of the others. Actually, looking at this, the way you kind of look at it, there's subject 3411 and subject 3408. It looks like they're kind of similar, at least compared to others. Maybe, okay, well, what's going on there? That's what's going on. Let's cluster the fused network and let's plot it. Let's start looking at these patterns of similarity, right? We have this matrix of fused similarities according to 3 data types. Let's figure out, are there subgroups of people? Can this be applied to precision medicine? What do they mean? Who are these people? SNF package has a large number of built-in functions to estimate the number of clusters that are given in a graph. You give basically a number of clusters to check. Here we're looking between 2 and 10, and then it's estimating basically how many clusters would provide a solution where the variability within clusters is kind of less than the variability between clusters. And we can run this whole block. So it's going to estimate the number of clusters. It's going to perform a clustering algorithm, spectral clustering, given a couple of options. So let's take a look at the first output. We'll go bit by bit here. We run estimate the number of clusters, and here is the R console output for that kind of estimate. These measures are eigengap and rotation cost, are 2 measures. There are a lot of different measurements for identifying cluster similarity. I didn't work in time is always lacking with this. We didn't work in a discussion on how to determine clusters, how to determine ideal numbers of clusters, how to determine cluster stability and cluster significance. Part of the gotcha at the end, it'll help you on your way. Suffice to say, eigengap is a very common one. Most of these measurements, as I say, are kind of based on looking at, if you take all of your individual observations, you take all these people that have been clustered given one iteration, one attempt. So here we say, okay, let's take a look at what happens if we give 3, a number of 3 clusters. We have to put every single person into one of 3 groups. The algorithm runs, we get 3 groups because we forced it to give us 3 groups. What we can do is look within those groups and basically get differences, and there's lots of different ways of looking at differences, of data distributions across what our input data type was between and within groups. Basically just looking at overlaps of distributions of kind of similar, we can use similarity, or not similarity network, principal components analysis for this. We can use this, this is why it's called eigengap. These eigenvalues and eigenvectors can give us information on how sets of variables within one group are aligned, or how they are kind of grouped into a latent space compared to the same set of variables in each of your other strata in your different clusters, and then compare the differences or the kind of the gaps in those calculations between groups. Another really useful way of doing this is with an alluvial plot, and I'll show you that. That's more kind of cluster stability. So what we got out of this was, a lot of this is automated. The best solution according to one method was 3. The best solution according to another method was 4. In heterogeneous data, there's never going to be a convergent solution. So we ran it both ways. That's what we did next. I ran a spectral clustering on the fused network with a 4 cluster solution and a 3 cluster solution. This is the result for the 3 cluster solution. So the patients now have been clustered according to those 3 groups, and you can kind of see here, these are the similarities, right? These are the values of similarity coming from that network W object. You can clearly see structure. You can see there's a group here that seems to be really similar according to the fused network, and a data set, group here that's pretty similar, and here pretty similar. Here's the 4 cluster solution. Is that as good? I don't know. There are ways that we need to validate it externally, biologically, clinically, and we're going to do that. So you could never tell by looking at these. There's no way to quantify the validity, certainly not the external validity of this clustering solution. And the attempts at establishing internal validity are almost always going to be conflicting. This is the alluvial plot. This shows each line is a person. And this shows on the left the 3 cluster solution, on the right the 4 cluster solution. This shows how people are reassigned depending on the clustering solution. So you can see, look, cluster 3 in the 3 cluster solution kind of gets broken up into 2 clusters. Cluster 1 gets broken up, although most of cluster 1 in the 3 cluster solution becomes its own cluster. Cluster 2, most of it. But cluster 2 becomes cluster 3 entirely. So we can learn things from looking at this and say, well, whatever cluster 2 is in the 3 cluster solution, it maps perfectly onto cluster 3 in the 4 cluster solution, and it's highly stable. Does not get split up, does not get broken. And we can look at the clustered object. It gives all of the IDs. So I've done another step. I've snuck in here. This is something also to consider with clinical data. You will have noticed in the wide format data set that we used, that remember that the phenotype data, pheno-wide that we used, the IDs were like sub 1 and sub 2 and sub 3. And then in the omic data that we fused, the IDs here, SNF ID, right, we're like 3406, 3407, 3408. This happens a lot. Often you have different sources of data for the same people in the same study, and the IDs are unique to the data type and the collection mode. And you need an ID key that links the two things together. So this step here is matching the data sets together. This is a file here, this ID key file, that contains one column with these SNF IDs and one column with the pheno IDs, and it lets us link. Now we can link our omic data and indeed our cluster definitions. So these are the cluster definitions that came from our clustering algorithm. I've just put them into data frame here. And I've merged in the SNF ID for our omic data and the phenotype ID that came from our demographic and our other imaging data and the cognitive data. Now we have a way of linking these. And in fact, now we've gone about this process of integrating EEG, methylation, gene expression into subject-centered information on who is more like each other in terms of overall biological electrophysiology, gene expression, molecular profiles, and created a new variable, which is cluster membership. We don't quite know what it means, but we do know exactly where it came from. We know the data types that made it. We know that they are biological and can be interpreted as such. So let's try to figure out what these clusters mean, right? Because then we can backtrack and we can try to identify. Does this represent a precision medicine model? Have we identified in one of these solutions a cluster of people that we could get from an EEG recording and a blood test, right? It's methylation, gene expression, and a cap, an EEG cap for a little while. Can we group people into clinically meaningful groups? Well, let's look at what we have. This is often what we would do next. We have a withheld set of variables. We have a model that we'd like to test. One of the key things we want to test is, as we did in our previous published work looking across diagnoses, how do these biological subtypes map onto typical DSM diagnoses? So let's take a look at that. What is the cluster overlap with clinical diagnosis? And we can do that. Basically, we're merging here. This is the merge step. We've got our phenotypic data that has diagnosis and age and sex. We've got our clusters, right? The three cluster solution, the 4 cluster solution, and we're merging them together, right? By our ID and by the pheno ID using our key, right? So we're merging the data together into an overall pheno file. We're creating a table that shows the overlap of diagnosis and cluster. So it creates a contingency table, who in which clusters belong to which diagnostic categories. We run a chi-square test on that, which is the statistical test for the independence of categorical variables. And then we have a plot. So I'm going to run all that and I'll show you what those outputs are. Here's our table. You can actually think of this in some ways as a confusion matrix, though we haven't kind of performed any learning, but we have made. We're not trying to predict our group memberships. We have come up with a new, and this is for the 4 cluster solution. And any of these steps, we can choose to substitute out. See, in this case, I've chosen cluster 4, right? But this data frame has 2 potential cluster solutions, right? Cluster 3 and cluster 4. Each person has been grouped twice. So this is showing the 4 cluster solution, which is nice because our data set has 4 different clinical diagnoses: bipolar disorder, dysthymia, major depressive disorder, and major depressive disorder with psychotic features. And now we have our 4 subtypes that we've identified with biological information. And looking at it, can we see any patterns? Is there any bias for different clusters belonging to different diagnoses? It can be harder to see in a 4x4 table like this. The chi-square test does it for us, tells us if there is independence between our columns and rows here. And the very low p-value would suggest that there is not independence, that it is not a random assortment between these clusters we've identified and our clinical diagnoses. And the plot that I've generated helps us visualize that. So here along the bottom in the 4 categories are our 4 clusters, cluster 1, 2, 3, and 4, biological clusters, and then the proportion of subjects is given on our y-axis, and they're colored according to the different diagnoses. And what we can immediately see is that, well, cluster 3, and if we remember, I believe it was cluster 3 that was highly stable between our 3 and 4 cluster solutions, seems to be actually incorporating almost all of the major depression with psychosis patients are in cluster 3. In fact, almost half of cluster 3 is made up of these patients, as opposed to cluster 1, where almost 75% are the major depression patients, or just the major depression disorder patients, and a fair number of dysthymia. There are no dysthymic patients in the third cluster. So, as you can see, we haven't identified an algorithm that can discriminate between diagnoses. We weren't modeling diagnosis. We've created something new, right? In fact, we could think of this more along the lines of RDoC, right? We've identified genes and potentially brain regions or brain circuits from the EEG, or brain signals, or electrophysiological signals that might represent specific features of a disorder as opposed to the diagnosis itself. 3 might be related to an RDoC construct of kind of externalizing behavior and psychosis, psychotic symptoms, hallucinations. So we can explore it a little bit further. Let's test a primary outcome. Let's say we were interested in evaluating the change in depression score between our 2 time points. Remember, we have longitudinal data, right? Each of these people, they might only have 1 time point for the data we used to cluster them with SNF, but they were measured at 2 time points for cognition. They were measured at 2 time points for some other variables like imaging. And so this exercise here is defining a new variable that's the difference in score between first and second visit for cognition, and then running a linear model, so linear regression. We're actually going to model this difference over time as a function of our 4 cluster solution, and we're going to add sex, age, and socioeconomic status. And we're going to evaluate statistics on that to see what the effect is. And we're going to do the exact same thing and compare its predictive ability with a model, the exact same model, but instead of using our biological subgroups, we're going to take the clinical diagnosis that's been given to them. So let's look at those 2, and I'm going to execute these blocks separately. It'll give the output below no matter what, but we can look at it one bit at a time. Here's the output from the regression model for the response of the difference in depressions over time. Because my font size is so large right now, it's making this table very difficult to read. It shouldn't be so hard to read, and maybe on your computer, if you're running the code, it'll be easier to read. The take home I want you to get, and without changing font size, it's going to be really hard to. This is the downside of R Markdown, its flexibility. What I'd like you to see, which is impossible because the lines have been shifted, there is a strong effect. It's hard to see because it's over here. Okay. 1.1 times 10 to the negative 5 is the significance ascribed to the beta coefficient for the difference of cluster 3 in contrast to cluster 1. These are all in contrast to cluster 1. This is how we interpret linear regression. I'd be happy to talk linear regression another time. But this visualizes it. Basically what we've seen is here's the change in symptoms. It looks like everybody in this study got better. Their change in symptoms was positive. They went up, although actually I think HAM-D is up is worse. So we can think about the organization of that. But most people in the study did fairly well. But this one group, cluster 3, had the opposite pattern, right? They almost all of them had a kind of below 0 change. So the biological subgroups here seem to be relevant to response to this variable, clinically relevant. Let's compare it to, I'll run the whole block because I get all those outputs anyways. We can compare it to how diagnosis fares if we look at the kind of classical DSM-5 diagnostic criteria and look at how individuals responded there. And here it is. Well, it looks like those with dysthymia seem to have an increase over time, but other groups, the majority of subjects, people in bipolar and major depressive group, didn't actually have as much of a change. If we look at the kind of ability of these algorithms to discriminate between people who are going to go below that line and above that line, the statistics, as they're supposed to reveal, this is a kind of a summary table that shows how well each of the models performed. The point is here we're evaluating if our biological clusters are statistically more significant, better at discriminating, in this case, a response versus non-response variable, people who did better or worse, which algorithm is going to produce more accurate groupings. And as I've engineered this problem to yield, the C statistic here for this model, which is the AUC, which is the discriminative ability of the model, is higher than the classical diagnosis. So in this case, we've got an AUC of 0.77. Here they've been multiplied up, so the C statistic is to the -1, so it is 0.77, and here it's 0.67. So there's an improvement in model performance here. We've done it, right? We've done it. We've used SNF and EEG and integrated with multiple omic data types to categorize people in a new way that is rooted in biology and modeled along with clinical and demographic data, age and sex, and outperformed the traditional symptom-based kind of symptom similarity measurements of traditional diagnoses. So, congratulations to us. Next stop, front page of Nature. Probably not front page of Nature. Someone will discredit it, I'm sure. The final step to this algorithm, that is kind of the final necessary step, is to go back now. So let's say we've identified these clusters, we have clustering solutions, we've performed a validation of those clusters against data that was withheld from our clustering, and we know that it's related to the response rate in our study, and we want to understand what was driving that. Which of the features that were fused in the network was more important? Was it EEG? Was it methylation? Was it expression? And within those, which of the variables caused people to be grouped similarly and therefore generated those groupings that were so informative for our study? And that's what this block of code does. It's nicely annotated for you. Most of this is ranking features. This ranked features by mutual information is built into SNF and does everything for you pretty much automatically. And it takes each data type individually. So we're giving it this list of data types that has all three of our data matrices in it and generating our rankings per data type. And I think it makes most sense to kind of go through. I'll run them all again because we can go through the outputs together, and I know that a little more than half of us aren't actually running the code. So it's going to rank the features. It takes a little bit of time. Again, we actually don't have a very high dimensional space here in terms of the number of variables we put in. So of course, as you start looking at thousands and tens of thousands of variables, this workflow will become more intensive and time consuming. But what we get out of it is a bunch of output from the console. So we get the ranking of expression, EEG, and methylation features. And we get a mapping that I've done just simply by kind of mapping the variable names from the original data sets back onto the rankings that are provided. For each data set, we have the range of informativeness of kind of mutual information that's given by all the features, the highest and the lowest. And then the ranking is obviously just going to be the highest, the lowest rank or the highest rank and the total number of features. And then the mapping of those onto each gene. And again, this is engineered to be this way. The most informative gene in the gene expression data set was SST, which is the somatostatin gene, which is identified as important in depressive networks by Itai's work and by single-cell networks in Alzheimer's disease, as well as in depression, as well as in a large number of other psychiatric illnesses, very important inhibitory cell type. The epsilon-2 parameter from a cognitive network from Andrea's work, kind of how we can estimate updating of beliefs and how we can kind of measure the activation, how we can kind of derive this feature from objective measurements of electrophysiology, and then this particular CpG methylation site. And you can confirm this if we just then derive a model after the fact of our, if we actually model this top response. So for example, now I'm taking this feature of just the most informative methylation site, and modeling it as a function of our clustering solution from the total network, it's an extremely significant model. It's very highly related, and actually part of engineering this is actually difficult to make all this work in a certain way. But it turns out that most of the clustering solution was actually driven by the CpG methylation at this particular site, and you can see that almost everyone it's almost a non-overlapping level of methylation in the third cluster for this particular. So the point is you can retrace your steps. You get these clusters, you have multiple data types, you can retrace your steps and find the individual features in your original data space that drove those groupings, and then explore them after the fact. So that's pretty much it. I was going to say if there's time, there's some nice network plots in R. There's not time. I've gone 60 seconds over, so I'm not going to explain any of it, but I'll run it just for the sake of running it. And again, a lot of this is just a resource for you to explore. And if you're hoping to kind of try some of this research on your own, it's a resource to explore. You can ignore all of these warning messages. There's a lot of that. And I should have put the quiet up here. If we reshape the network object, so again, all of this is annotated. Let's just take that similarity network that we had and get a nice view of our similarity. We already had those kind of matrices where we had blocks of patients and the dark and light spots, but we can use these packages to generate a nicer, maybe more intuitive kind of spatially organized network where our 4-cluster solution is represented here. And as you can see, something that's revealed here quite explicitly is our cluster 3, all the individuals that were clustered into cluster 3, you can see how closely related they are, right? The thickness of these lines represents the similarity between observations, and you can kind of see the overlap between the remaining clusters, right? 1 and 4 and 2. So this is a 2-dimensional representation, network representation of this multidimensional kind of scaled network. And the gotcha, which isn't really a gotcha, someone has gone and developed a package that integrates not just SNF, but several of those other multidimensional, multi-omics approaches. We can do everything we just did in these steps, not everything, but at least running the SNF we can do in 2 lines of code. If we use the package called cancer subtypes, this is kind of the last point. So the gotcha is check out this package because it'll make your life easier and also provides a lot of built-in tools for checking the validity of these clusters and the stability of these clusters beyond looking at eigengaps and silhouette plots and alluvial plots and things like that. Really great package. But take home here, another take home, profound point. The package is called cancer subtypes. There is nothing unique to cancer about anything we've done. There's nothing tailored for cancer that these methods do. There is absolutely no reason why we brand some statistical methods the way that we brand them. Here, you would not know about this package if I hadn't shown it to you, or if you hadn't kind of gone looking for a way of implementing this specific methodology. SNF is not exclusive to cancer. This is siloing, right? And it's broader than just psychiatry. Right? This is like biomedical science. People think methods applied in cancer or cardiology, looking at peripheral illnesses, looking at anything that's not mental health or addictions, which is still kind of, looked upon. There's still stigma in this, in the research world even. People, it's almost too messy, too difficult to deal with. There's no reason to think that the methods are different, that the numbers are different. We're looking at gene expression, we're looking at tissue, we're looking at biological processes that drive complex phenotypes. So don't be afraid to look beyond the canonical resources for methodologies that can be useful to gaining insight into what we're trying to look at. So that's about it for me. On that note, we're 5 minutes into lunchtime. I do want a little tiny bit of housekeeping if you'll indulge me for the next couple of minutes. I really want to thank Erin, our fearless leader, for all the work that she's put in, not just in the last week and a half, but leading up to it, putting on the just organizing everybody, all the TAs, all the stubborn PIs and scientists and postdocs, everyone who's going to do something. We all have other things going on. She has other things going on. She's got a family. She's got tons of projects underway, and she found the time to build the Docker, to build a binder, to work on the GitHub and organize everything, make everything work, double check everything. She's been updating all of these resources like 24 hours a day, communicating with all of us. So I just want to give a round of applause to Erin, who is the blood in the veins of this entire summer school. And so with that, I'll wrap it up. I know there's a couple of questions, but again, I'm counting on. Actually, maybe I should write them down. I'm counting on whoever put the question in the question box. I know Amanda's asked a couple questions here. Please don't forget, if I do forget to ask them. I thought Shreyas might be logged in and looking at. Maybe Shreyas is doing this. Yeah, actually, Shreyas and Kevin, it's on you. We're going to answer all of the questions that have been asked in the afternoon and more. So remember, bring your questions. We'll come back at 1 o'clock. And we're going to have just a nice discussion about broader topics. We can even delve in specifically. Let's talk about training. Let's talk about academia in general. And we really hope you've enjoyed all of the material. Please fill out the surveys for how much you enjoyed or didn't enjoy the course, things you'd like to see changed. And it's really important to all of us, including Erin and everybody else who's taught and organized this. So please fill out those forms, even just in the minimal way if you have no time. And we'll see you in the Gather Town as well, if you'd like. So there's the post-workshop survey in the chat, and we'll kind of try to chat in the Gather Town over lunch and come back for a broader panel discussion. So thank you all very much, and I really appreciate your time and attention.